Hey guys, it's Karen from GSA. This is our first review video that we've ever done, so in place of having a review session, we are going to do this for this exam at least and see how it goes. We figured that it might be easier for you guys to not have to make time in your schedule to come all the way out to campus if you weren't already there, or if you have to work, things like that. We definitely wanted to try and accommodate you guys. So what I want to start off with first is give you guys a little bit of an idea of what the exam is going to be like, what to expect. So as you guys know, there's not a lot of calculations for this portion of the exam, or for this portion of the semester. There aren't a lot of calculations at all. My phone is going off, I'm sorry. Yeah. Okay, so there aren't a lot of calculations. You've got mostly conceptual things. So you're probably thinking, the multiple choice is going to be very straightforward, but what is the short answer going to be like? And my answer to you is that it's going to be mostly case scenario based. So you're going to be looking at a lot of things where he's telling you, this is the data we're trying to find, what's the best process? Or if we miss this step in the process, or if this isn't added in the process, how will it be affected? How will the end result be affected? Another type of question he could ask is something more pedigree related, not necessarily having a pedigree where you need to figure out any probabilities, obviously, nothing like that, but just saying something along the lines of mutations. So what mutation could explain this? Could imprinting have caused this? If so, why not? So that's why it's important to know the different kinds of mutations, to understand what imprinting is, and different things like that. So that's the best insight that I can give you on the short answer portion. You know, there's not going to be calculations. It's mostly going to be you explaining a certain concept that he's presenting to you with the knowledge that you have. A good thing about this packet is that it includes all of that information, so you're going to be more than prepared with that information for the exam. So I want to just go ahead and get started. What I'm going to do is go page by page through the packet. I know you guys are super smart and you don't need me to read it to you, so I'm not going to do that. I'm just going to go through, keep this video as short as I possibly can. I don't want to take up too much of your time, and highlight the important topics that I feel are going to be most important and most pertinent for you on the exam. Don't forget to check your emails. I just sent one out for everyone who has already purchased the packet as of tonight, March 17th. An edit to number 7 on your practice problem sheet. I left out a little bit of information that you definitely need to solve the problem, so be sure to check that. Alright, so let's get started. So we'll skip the first page because you guys already know that. We start off in lecture 10 talking about the DNA binding proteins. Definitely read through this page and understand it. It's a lot of just definitions and kind of basic stuff that you already know. You reviewed activators and enhancers for the last exam. This goes in depth specifically for enhancers about Cathari, which was a scientist. He came up with, he did an experiment on mice. Just kind of, that would be a good true-false question at most, but since he talked about that scientist, I would definitely be familiar with the work that he did, and that's the last bullet point on this page. There's also the sonic hedgehog gene, just knowing that that's an activator. I don't know if he's going to throw that in as a, just a freebie true-false, but that's all I can really think of for those experiments specifically. Taking a step back and looking back at the earlier stuff on the page, the helix loop helix transcription factors, just showing you the mic, the max, the max, the mic, the mixed. Those are confusing. So MAX, when it's bound to another MAX, there's just basal transcription, and all that means is just a very, very low level. And then you've got when MYC is with MAX, you've got very high transcription, so that's favorable. And then lastly, when you have MXD with MAX, you have just no expression at all, so it's inhibited. I would definitely be familiar with those 3 specific gene transcription factors and what they do, or how they affect transcription levels, I'm sorry. And I believe That's all that I would really suggest knowing very well on this page. Definitely read through everything because you do need to know everything in this packet. But as far as things that you need to have a good grasp on, that's what I'm going to go over, just to be clear. I don't want you to think that I'm only going to go through and highlight the things that you need to know. You need to know everything in the packet, but these are the things that I feel you need to know the most. So the LOD score, I'm going to go back to that really quick. It's under the third bullet on the page. Logarithm of the odds is what that stands for, LOD, logarithm of the odds. Generally, if you have a LOD score of 3 or more, that means that 2 gene loci are very close to each other on the chromosome. I would just know that because it's a very easy thing for him to tack on to a short answer, saying that a LOD score of, these 2 genes was 4. And, what does this indicate? And then you would easily know that, okay, well, it's 3 or more, so these genes are very close to each other. Okay, so let's move on to page 3. Enhancer traps. Definitely be familiar with how that works. I've included a diagram here that you guys can just read through, and it really just shows you everything you need to know regarding that. OCA2 and HERC2 are 2 specific genes that he talked about in class. Because he took the time to single out these 2, I would definitely know their functions. I think that that's an easy question to get right. Especially OCA2, because we're about to talk very much in depth about the tyrosine transport pathway, that type of a thing. So I would definitely know OCA2 and how it affects pigment protein in the eyes. Moving on to CHIP sequencing. You don't need to know the exact, maybe that would be just like a freebie question. So if you want to know that it's chromatin immune precipitation sequencing, then you can be fancy and you can know that. But more importantly, you need to know what goes on and why it is an important process and what we can learn from it. So moving on to the next page, there's a great diagram there that shows you exactly how the process works, and it also shows you what chip sequencing is used for. So the most important thing you should take away from it is the first bullet point at the top about how often a sequence is seen on a map indicates how strong the protein binds to it. So if you see a high peak on your genome map that you receive from the chip sequencing, then you know that the sequence was seen very, very frequently because of that high peak. And you also know that the protein binds very, very strongly to that sequence. MSX1, I don't think you need to know too much about this gene. The most I could see it being used on the test is as a simple trick multiple choice answer. So if he asked what chemical could you use for a Western blot, which he didn't go over that, so he won't ask that, but I'm just saying as an example. And he included a bunch of chemicals, one would be the right one, and he might tack this on as, MSX1 as an answer choice. And as long as you know that what it is, which it says it right here, then you would know that that wouldn't be a valid answer choice. So knowing that would just be a great way to narrow down answer choices, but I don't think it's something too pertinent for you to know. Moving on to the trip operon, this is what I was talking about earlier. We know from previous exam that tryptophan acts as a co-repressor, so the trip operon itself is always transcribed unless tryptophan is present. And then once tryptophan is present, it binds to the repressor. It has a little spot just for it that it binds to. It changes, the repressor changes conformation with tryptophan bound, and then both of them together as a unit bind to the operon, and that's what shuts off transcription. Okay, so 100% expression is not always observed in the trip operon in general, not usually ever observed, but more importantly, if we took that repressor away permanently, if it was broken, if there was something wrong with it, and it wasn't functioning, we still would not see 100% expression. We would only see 33%. Not something important to know because he mentioned the study in which that was found, so know that. I think that's an easy true-false question. know that if you took the repressor away, there would not be 100% expression. It would only be 33%. Okay, moving on to the next page. This is an important page to read through and to understand. I'm not going to go through all of it. I'm just going to hit the highlights, so the main points. Attenuation is a very important concept to understand. The first bullet addresses exactly what it is and kind of what it does. It decreases gene expression. I was going to say it's a repressor, but it's not. It just decreases the expression of the genes 700-fold. So you can imagine that that's a great amount of repression going on here, of limited gene expression that's happening here. So this just kind of goes into the process of how the hairpins form. The different ones are 1, 2, 2, 3, and 3, 4. 1, 2 is rarely ever formed because RNA polymerase waits for a ribosome to attach before it continues that translation past the first sequence 1, so that rarely ever happens. But when high levels of tryptophan are present, the 3,4 structure forms and RNA polymerase dissociates from the DNA. That is important to know. Definitely know what happens when high levels of tryptophan are present. It's an easy multiple choice question. Even a case scenario, he may ask something like that. He may show you a graph showing tryptophan versus another type of thing, time, environment, anything. And this would be a great thing, a great resource to have in your mind to be able to answer certain questions. When low levels of tryptophan are present, the leader peptide, so you'll read more about this in this paragraph, and you'll understand what the leader peptide is. Basically, at the beginning of the leader transcript, there's about 14 amino acids that are called the aminopeptide, and so if a ribosome attaches and attempts to translate during low levels of tryptophan, it'll stall at either of the two tryptophan codons, which are also explained here. And then while it's stalled, it shields the 1 sequence and the 2,3 structure forms. Alright, moving right along. Okay, so here's just a diagram on page 6 of the high level of tryptophan, the low level of tryptophan, just to give you an idea of, what it looks like. And then above that, it says what would happen if you had a region that deleted region 4, which is the region that determines termination. And this was an example that he used actually in his slides. A question that he used, and it says, and so the answer is that termination wouldn't be able to occur because you can't form a proper secondary structure. Post-transcriptional control. Okay, this is where I really wanted to kind of tell you guys that this is something that you absolutely need to know. These different types of control, again, I'm not going to read these out because they're very straightforward in here, but I'm going to list the ones that you definitely need to know, okay? Which is going to be most of them. So, riboswitches, alternative splicing, RNA interference. Put 2 stars next to this one. Definitely take the time to read through this because there's a lot of information and a lot of potential for a short answer question. Moving on to the next page. DeVos et al. That experiment he took the time to go in depth in class. He even, showed you the procedure that these men did, or these scientists did, when they did their research on the tau protein to see, so the tau protein, if you don't already know, influences a lot of degenerative brain diseases, including Alzheimer's and many others. And so in the tests, in young mice, they found that by eliminating this tau expression, it promoted learning and memory, and it also reduced the incidence of seizures, which was pretty impressive. It was unclear, however, if reducing this expression in mice would be just as effective, and so they sought out how they could do that. That was the purpose of their experiment. So definitely take the time to read through this. It's very interesting, and it's also fair game to be on your exam for sure. Gel electrophoresis, you guys have talked about this a bunch of times. I'm on page 9, by the way. Just kind of a review section there to, things to remember for the exam. And then we get into blotting. So just know the different types of blot testing. So Western blot, Southern blot, Northern blot. That the Western is protein probes only, so it's protein specific. It's the only one that does just proteins. Southern blot and Northern blot are both for nucleic acids. Southern blot's for DNA, Northern is for RNA. And then QRT-PCR, I would just read through this and know the process. I think that's the only thing you need to know about that is just the process, and that's the second bullet. It's also good to know what it helps scientists to do, so it enables reliable detection and measurement of products generated during each cycle of the PCR process. Okay, moving right along. So now we're at in situ hybridization. Again, just read through this. Definitely something to know. Okay, and now let's move on to lecture 12. So chromatin organization in DNA. This is also something that you've definitely gone over in other classes about how chromatin is, formed with the histones, and it helps to condense the DNA because we know how long and huge DNA is, and it would be chaos if it wasn't condensed in some kind of a way. Heterochromatin, know that that's tightly packed, and know that euchromatin, EU chromatin, is the loosely packed form. Moving on to the last bullet of this page, chromatin remodeling, know the two types of promoters, the open and the covered. And those are explained on the next page, page 11. Definitely read over that and know the differences between those two. DNA methylation. This is something that's also important to know. We're getting right into the meat of what you guys need to know right here about how the 5-methylated cytosine is present a lot of times at the CPG islands. So definitely read over this. It also explains bisulfite sequencing, which is something that you also need to know. So you could probably highlight that. Histone modification. This is a section that you'll only see in multiple choice. I could see this being 2 multiple choice questions. So definitely take the time to read this. HAT, histone acetyltransferase, it acetylates lysine amino acids on histone proteins, which means that it increases gene expression. Know this. Highlight this, star this, whatever you need to do. This is very important. And you also need to know that deacetylase decreases the gene expression and therefore turns off genes. So just know the difference between those two, know their names, definitely be familiar with them for the exam. Epigenetics, this is where we get into imprinting. Main things you need to know about imprinting, you need to know everything that's written here, so definitely read through this. But what I want to do is actually just show you that under the third bullet where it actually talks about imprinting, past where it says certain genes are expressed in a parent of origin specific manner, it says if the allele inherited from the father is imprinted, it is thereby silenced, and only the allele from the mother is expressed. If the allele from the mother is imprinted, then only the allele from the father is expressed. So as long as you know that imprinting influences which parent's genes will be expressed over the other. So that is the big fact that I would know, if anything, and all the little details that are also listed here definitely should know for epigenetics. So very important topic to know. And again, the imprinting slides that he had were really, really great. He talked about Prader-Willi syndrome and Engelmann syndrome. which are mentioned in the second slide that I put on there. Highlight that. Know the characteristics of both of them. Not in depth, but just how they differ and how they're inherited. Insulators, he doesn't go into very much depth on his slides on this, but you do need to know these. So they influence promoter and enhancer interactions. That's a very important key element of what insulators do in general. They usually block the interaction between the two, and they determine the set of genes that an enhancer can influence. So just read over that, know that. Okay, so X inactivation, exist RNA, has a lot to do with bar bodies. So it's basically the long non-coding RNA. It is the bar body essentially. It's the X chromosome that is not expressed. And this just shows you exactly the key. It's exist RNA is the key material that is a part of this inactivated X chromosome. So read that, definitely know it. I have a practice problem in the practice problem set that addresses this concept as well. So definitely know at length what it is. Moving on to lecture 13, we have DNA mutation and repair. I am not going to go into too much depth on this lecture just because everything in it you need to know. So everything on the next page of, lecture 13, you need to know. The Lurie and Delbruck fluctuation test, I would know at length what they discovered. So, read through this and know how, if the assay that they were able to develop from their mutations, research, things like that. And then we move down to the last main bullet point, so what causes changes to DNA. This is where we get into the different types of mutations, and the list goes on. So if you haven't started looking at these, a lot of these you know, so not to overwhelm you, because a lot of these you do know, but make sure that you refresh them in your mind, that you understand exactly what they are. I think you probably know the first few. Spontaneous error may be new to you. You probably know missense, silent. Strand slippage is a very interesting type of mutation. I've put a diagram there to help you kind of see that, especially on test day when you're just trying to brief through them, and in case you need a little bit of a visual, I've included that for you there. Again, it can happen on both the newly synthesized strand and the template strand. One is not favored over the other when it comes to that type of a mutation. Page 17, we've also got many, many more different types of things that can cause changes to DNA, different types of mutations from loss of function to gain of function to, UV light, radiation, x-rays, everything. So just be sure you know all of those and the consequences that occur as a result, because again, case-based scenarios are what you're going to have for short answer. Also for short answer help, look at your recitation section, session problems, because they're very hard, I've heard. I haven't gotten a chance to look at them, but I know that they're pretty difficult, and I think that's a good thing that he did that, because it means that his exam questions are going to be similar. And so if you practice those a little bit, then you'll get a good grasp of what to expect on the exam. Whether or not they're similar, you know that they're going to be at least at that level or even a little bit easier, because again, it's concept-based. This whole exam is concept-based instead of straight calculations. Okay, let's move on to page 18, because again, you guys can just read through this. The Ames test under identifying mutagens. Know what this does. So know that this uses bacteria to detect whether a given chemical can cause mutations in the DNA of an organism. Just know what it's used for. Let's see. How do cells protect themselves? Moving down to the bottom, definitely look over mutation rates. Again, like I said, everything in lecture 13 is important. Everything in all of them is important, but especially lecture 13 when you've got these mutations and how DNA repairs itself. It's very specific stuff. It's not broad the very specific. So know these for sure. These types of proofreadings, you should also know the direction in which these proofreadings occur. A lot of them are in the 3' to 5', but there are some that are 5' to 3'. And then at the bottom of page 19, I've included a slide from Dr. Armstrong's lectures where he talks about DNA repair and disease and how these different types of DNA repairs can cause certain diseases. So definitely take a look at that because it's interesting, and again, it's fair game for the exam, easy multiple choice question. Okay, site-directed mutagenesis, just take a look at that on your own time. We're going to move on to lecture 14. I want to say something brief about this lecture really quickly. A lot of it, I know that his slides can get a little bit confusing at times, or it seems like there's not enough information. For lecture 14, I direct you, I think twice, back to his slides to go look at the information he's presented you with, because there isn't much else I can say, if that makes any sense. So take it for what it is. Do not overthink it. I guide you in here on exactly what to look at, what you're looking for, and I show you which slides you need to study specifically to kind of help you out with that. So here we're looking at double strand breaks, and actually a lot of what's in lecture 14 deals with double strand breaks and how they affect DNA, how they're caused, different things like that. As you can see on page 21, I've included a lot of his slides because I think they are really helpful here. I think that they have a lot of information, a lot of the same information, especially that I would have told you to try and help you understand these types of things, and I think that his slides do it best, right, for lecture 14. So definitely go back to it. Take a look at some of these slides, recombination repair especially. Those are the slides that I would focus on. This evidence of the double strand break repair during meiosis, something, just to keep in mind that there is repair during meiosis that occurs, because that could easily be a true-false question, multiple choice question as well. Okay, know what a heteroduplex is, if you want to underline that or highlight that at the bottom of page 21 as well. Okay. And page 22, almost to the end. I promised you guys I would keep this short but informational. Again, if there's anything else that you guys would like to hear about, if you want me to email you personally, again, send me an email, ask me your questions. I know your test is on Thursday, but I'm here to help in any way that I can. Okay, so antibody production, I guide you to slides 13 and 18 here, because I think they have a lot of information, a lot of diagrams that may be helpful to you, and moreover, they cover the meat of the information that he's going to have in his exam. Again, it's mostly just for the diagrams that I'm sending you to these slides. You should definitely know gene knockout and gene knock-in. I've included that here, even though they're on those slides. The information that I have under this bullet point is included in the slides that I direct you to, but I put it on here as well because it's what I want you to kind of keep in your head as you're flipping through, on test day, and you're looking for things to kind of just remember, to just click and things like that. These are the things that you should remember. The CRISPR-Cas system, definitely read through this. Know that this is a bacterial adaptive system that protects against viruses and promotes recombination. Know this information. Know what CRISPRs are. Know what this system, how this system works. Not necessarily what it does, but how it works. And that's it for this review. I know it was very short. Again, if you guys have any specific questions about things that are in the review packet, things that you wanted me to go over a little bit more, please let me know. Again, this is a new thing that we've tried, and we felt that it would be better for you to be able to watch on your own time. So let us know your thoughts, and again, email me if you need to. It's KDSOUZA113@gmail.com. Good luck and happy St. Patrick's Day.